Good day, my name is Alfred Dayap. My topic is about biomagnification. What is biomagnification? It is a process which toxic chemicals transferred to its predators. It affects most the entire food chain. The animals that highly impacted of biomagnification is the animals who are in the highest position of our food chain. They are most likely impacted because they gather all the toxic chemicals by consuming their prey. Cause of biomagnification is through the chemicals and pollutants, example the heavy metals and pesticide. The process of it is through the absorption of toxic chemicals by our primary source, which is the soil and the water system. They provide the plant and many aquatic animals, leading to biomagnification because of consumption that build up to their predators. Biomagnification considered bad because it increases the harmful chemicals and substance in our normal process of food chain that resulting or leading to many living things will die. Even though it is considered bad, but it is important in some study like in toxicology. They considered or it is very important to them the biomagnification because it helps them provide the data the exact amount of pollutants in their samples. That's all. Thank you.
Good day, I'm Harold John C. Hampang and I'm gonna discuss about bioaccumulation. What is bioaccumulation? It is a process where the cell performs a selective absorption and storage of variation of molecules. It allows them to absorb nutrients and essential minerals for them to survive, but at the same time it allows them to absorb harmful substances as well. Toxins that are instead deleted in the environment can also reach dangerous levels inside the cell and tissue through this process of bioaccumulation. Bioaccumulation occurs when an organism absorbs a substance at a rate faster than at which substance is lost or eliminated by catabolism or exertion. Thus, the longer the biological half-life of a toxic substance, the greater the risk of chronic poisoning, even if the environmental level of toxic are not that high.
Good day. I am Elmar Cris Pesado and today we will discuss about measuring toxicity. When measuring toxicity, lethal dosage is what we will usually refer to the amount of a substance that will kill a specific percentage of the population, usually within 4 hours of ingestion. For instance, if the LD is 50, it means the population will die within 4 hours. If you have 10 mice, 5 of them will die at the end or by the end of 4 hours. And if the LD is 20, then 20% of the population will die after 4 hours. So if you have 10 rats, the 2 of them will die at the end of 4 hours. This means the higher the LD, the more lethal it is. Now let's talk about lethal dosage. Lethal dosage is recorded in milligrams per kilograms of body weight. We will estimate how much will kill a human based on lab rats of the results of how much killed a population of rats. This can be very difficult. Resistance can be very different among species. It can vary between humans and rats. The toxicity classes and definitions: if it has extreme toxicity, that means a taste to a drop could kill a 150-pound adult human. If it's relatively harmless, that means that it's more than a quart that will kill a 150-pound adult human. This means the smaller the amount, the more toxic the substance is, and the larger the amount of the poison that it takes to kill a person or a group of rats, the less toxic the substance also is. This table involves some lethal dose values, so some materials of some poisons may be very poisonous. Some of it may take a lot to kill a person. So, really, what it means is that the poison is in the dosage. There are some common things that humans will come in contact with. The exception of the top 5 most poisonous on this chart, everything else is naturally occurring of something that we may in contact with. Sucrose or sugar can actually kill a person in a very large amount. Same thing with water. Humans overdose from water periodically, but they actually have to drink a large amount of water in order to overdose on it. The top 3. These 3 right here are the most poisonous products out there, the most poisonous material on Earth. All 3 of them are naturally occurring products. That means that they are not man-made. They are naturally found on the Earth. Some of them are bacteria, some of them are funguses, but they are naturally occurring. Botulinum toxin right here is the deadliest form of poison. It is something that we use in medicine. It is what Botox is made from. We use a very diluted amount because it doesn't take much of this toxin to kill a person, so we use less than what is used to kill a person. We use it to paralyze muscles of the skull and face so it decreases migraines and also wrinkles.
There are 4 steps in risk assessment process. First is hazard identification. Second is exposure assessment. Third is dose response assessment. And fourth is risk characterization.
First, identification of hazard. It is used to evaluate if any particular situation may have the potential to cause harm and consists of testing materials to determine whether exposure is likely to cause health problems. One method used is to investigate population of people who were exposed previously, for example, to understand the toxicity of radiation produced from radon gas. Second, dose response assessment. This next step involves identifying the relationship between the dose of a chemical, therapeutic drug, pollutant, or toxin and the health effects on people. Some studies include administering responsibly high doses of compound to animals. The result of exposure will be different illnesses and symptoms such as tumor or rashes development are recorded for varying doses.
Third, exposure appraisal. This step evaluates the duration, frequency, and intensity of human exposure to a particular chemical, pollutant, or toxin in the environment. It includes some discussion of size, nature, and types of human population exposed to the agent. The total population exposed to the agent is directly proportional to the hazard in the society. Fourth, risk characterization. The final step aims to delineate health risks in terms of the magnitude of the health issue and concern that might result from the exposure to a particular pollutant or toxin. It is necessary to identify the hazard or danger, complete the dosage response assessment, and evaluate the assessment as outlined. This method involves all the uncertainties of the previous action and the results are again like to be controversial.
Those are the first step of
risk assessment processes.
thank you.